Hello, everyone, and thank you for attending today's BD FACSMatters Ask the Expert webinar on decreasing single-cell multiomics sequencing costs. I am Annalise Barnett, Associate Marketing Manager at BD, and I'll be your host for this webinar. Today's speaker is Stefanie Mortimer, Senior Director of Single-Cell Multiomics at BD. Prior to joining BD, Stefanie led the technology development and engineering teams at Guardant Health. She was the first scientist to join Guardant Health and built the technical team from the ground up to develop and transfer innovation into products and processes related to liquid biopsy technologies. Stefanie was a Leukemia and Lymphoma Society postdoctoral fellow in the lab of Professor Jennifer Doudna at UC Berkeley. She received her PhD in biological chemistry from the University of North Carolina at Chapel Hill and a Bachelor's of Science in Chemistry and Biochemistry from McMaster University. So before I hand over to Stefanie, I would like to go over a few housekeeping items. If you have any questions during the webcast, please submit them through the Q&A widget of your console, or technical application scientists will be answering them on the back end, and after the presentation, we'll be having a live Q&A session. For the best viewing experience, we recommend using a wired internet connection and closing any programs or browser sessions running in the background that could cause issues. Some networks cause slides to advance more slowly than others, so logging off your VPN is recommended. And if your slides are behind, pushing F5 on your keyboard will refresh the page. You can find additional answers to some common technical issues located in the Help Engagement tool at the bottom of your screen. Lastly, a recording of this broadcast will be available after the presentation, so look out for an email with additional details on where to access it. And with that, thanks once again for joining us, and I now hand over to Stefanie.
Thank you, Annalise, and thank you everyone for joining the webinar today. I'm really pleased to be able to tell you about the work my team has done to test multiple approaches to decrease sequencing costs while efficiently distributing sequencing reads for simultaneous protein and mRNA quantification at the single cell level. I really hope you get something out of it and you're able to apply this in your next single cell multiomic experiment. Workflow, as I've shown on this slide, is an extremely powerful tool to have applied to your experiments. From our perspective, it often starts at flow sorting to isolate your cell populations of interest. It then involves upfront cell preparation, which may involve using oligo-conjugated antibodies for protein detection or sample multiplexing. Next comes single-cell isolation and molecular barcoding, followed by library preparation, which can involve whole transcriptome amplification, targeted RNA-seq, and recently we have added the ability to do T-cell and B-cell receptor sequencing. After sequencing, there are several bioinformatic and data analysis steps to take the raw sequencing files and turn them into biologically meaningful data. We also see further validation on flow as a follow-on to confirm a certain cell population of interest that may have been identified as part of your single-cell workflow. We see the need to analyze both RNA and protein in a multiomic approach on the single-cell level because it can provide complementary data due to the inherent differences in protein and RNA expression and the regulation thereof. Here I'm showing an example where CD3 surface marker protein may be highly expressed, but its corresponding mRNA transcript in the cell is expressed at lower levels. Proteins have been examined on the single-cell level through fluorescently conjugated antibodies, then visualized and analyzed through flow cytometry. However, this approach, while it has a high cell throughput, is difficult to multiplex and does not allow for simultaneous genomic or transcriptomic analysis. Recently, the use of oligo-conjugated antibodies has allowed for readouts through massively parallel DNA sequencing. What this does is allows for simultaneous readout of protein and RNA or DNA and to have higher plexi protein panels applied in your cell experiment. The one caveat is that current single-cell methods are limited to throughput of only tens of thousands of cells per experiment. And also, the more analytes you measure, the more sequencing read depth you need, which can ultimately increase the cost of your experiment substantially. We also believe can be achieved with a single-cell multiomic approach comprised of both RNA and protein analysis. Cellular biology And specifically, the immune system consists of complex gene regulatory networks at the transcriptional and post-transcriptional level. Protein-only or RNA-only analysis can greatly limit the understanding of cellular heterogeneity and regulatory networks during complex responses. Protein profiling methods using oligonucleotide conjugated antibodies, such as BD-AbSeq on the BD-Rhapsody cell analysis system, can address this issue by allowing simultaneous analysis of mRNA and high-plex protein panels from tens of thousands of single cells with a single readout, improving the resolution and insights of your single-cell experiments. We have shown previously that AbSeq correlates very well with traditional flow cytometry approaches, so you can trust it to use in conjunction with your flow data. While detecting new molecules from single cells can open up new avenues of research, it can be costly to sequence deeply enough to obtain the reliable data that you need, and also most of the cost can actually go to only a few highly abundant targets. So to address this issue, we developed and tested 3 approaches: upfront T-cell enrichment, removal of high expressors, and also signal dampening by mixing unconjugated antibodies with high expressors, which I'm going to go into detail describing for the remainder of this presentation. For the Rhapsody single-cell platform, which I'm showing here, utilizes microwells to capture single cells and a single bead together in a single well. And this bead is precisely designed to fit in this well. The cell capture bead also contains oligos with a poly-T sequence to capture polyadenylated mRNAs from lice cells. The beads are also magnetic and allow for a simple retrieval step post-cell lysis and poly-A capture. The captured mRNA onto the beads by a reverse transcription step and the subsequent creation of cDNA. This also adds on universal sequences designed for downstream library amplification and generation, and cell labels and unique molecule identifiers, which are needed for the identification of putative single cells and the related molecules downstream, which is done in the post-sequencing analysis. A benefit to this encoding on the beads is that these can actually be easily archived and stored for later use. BD-AbSeq uses antibodies to simultaneously analyze mRNA and protein with high throughput sequencing. Similar to mRNA, the oligos conjugated to the antibodies that have bound that specific cell also get captured on the bead through the poly-A tail. The DNA oligo conjugates contain a specific barcode for antibody identification and a common sequence to allow for PCR amplification downstream. These all similarly get encoded onto the bead next to the cell label and the UMI as with the mRNA molecules. In this study with the BD sample multiplexing kit, also referred to as SMK, it is used to multiplex different samples onto one Rhapsody cartridge. These kits use a universal antibody and up to 12 unique sample-specific barcodes to allow up to 12 samples to be loaded onto one cartridge at a time. The bioinformatics pipeline then automatically performs demultiplexing downstream of the sequencing run, so you can identify each sample. As I previously mentioned, in this study, we assessed 3 different methods for reducing sequencing costs while getting the same performance for simultaneous detection of RNA and proteins at the single cell level. For the control in 2 of the methods, we used human PBMCs, and for the last condition, we used T cells enriched from this same PBMC sample. In all conditions, we looked at both resting and activated states of either the PBMCs or the T cells. In the control, we applied a 20-plex T cell panel under standard conditions. And in the first experimental condition, we signal muted the top 5 expressors in this 20-plex panel, which has CD45, CD11A, CD18, CD4, and HLA-DR. And we did this by mixing unconjugated antibodies to the oligo-conjugated antibodies in a 2-to-1 ratio. In the second condition, we just removed those same 5 high expressors, leaving 15 antibodies, which we herein refer to as the 15-plex panel. And in the third condition, we enriched for the cell type of interest up front, in this case T cells. T cells were enriched from PBMCs using the BD-IMEG human T lymphocyte enrichment set. The cell activation that I mentioned was done by CD3, CD28 for 24 hours. And ultimately, to distinguish the activated cells from the resting at each condition, we used the BD single-cell multiplexing kit. Cells were pooled onto and loaded onto one cartridge for each condition. Here is T-cell BD AppSeq panel design that was employed in this study. The first 5 specificities listed here are the high expressors that were either muted in the first experimental condition I described or completely removed in the second experimental discussion that I described, leaving the 15-plex panel. The next 10 in the list are the T-cell proteins that are implicated in activation, and so will be used to look at the differences between the resting and activated states in this experiment. And the last 5 are cell lineage markers that we included. Attribute we assessed post-sequencing analysis was the read distribution among each antibody in the panel for the control in each of the 3 conditions that we tested. In the control, which is shown in the top left pie chart, 68% of the sequencing reads were mapped to the 5 highest expressors, CD45, CD11A, HLA-DR, CD18, and CD4. So you are left with 32% of your reads mapping to the other 15 antibodies in the panel. However, when you mute those 5 high expressors with unconjugated antibodies, now only 9% of your total sequencing reads are mapping to those same 5 antibodies, and so you get the remaining 91% of your reads for the other 15 antibodies in the panel, and that's shown in the top right pie chart. In the bottom 2 pie charts, on the right, is the enriched T cells, for 57% on the sequencing read mapped to those same 5 antibodies because of the removal of certain cell populations that would be expressing those cell surface markers. So you do get a decrease, but only slightly. And in the 15-plex, which is the bottom left pie chart, the reads get redistributed over those antibodies. So right off the bat, this is very elegantly showing, before any further analysis, that our conditions would work to enable the reduction of sequencing costs while maintaining performance. In parallel to the Plex AppSeq panel, we also performed whole transcriptome analysis, or WTA, on these samples for all of these conditions. 8 total conditions were done when you consider both the resting and activated conditions. So here are the TSNE plots derived from the mRNA alone for the PBMCs across all the conditions, showing the segregation of cells according to cell type and status. What we observed is there are clear differences in expression between the resting and activated PBMCs, which are colored in red or blue in the different TSNEs. However, when you look at the control versus the experimental conditions, you see that they are very identical. So here we confirm that the WTA data is not affected by the different conditions we tested with the AppSeq panels. And antibody sensitivity. And so on the left-hand panel, we show heat map of the mean molecules per cell of the 15 antibodies that are consistent across the control and the 3 experimental conditions. And here, all the samples have been sequenced with the same number of total reads per cell. The first column in the heat map on the left shows the control condition. So if you looked from left to right, you can see the shift from more to more greens and reds, indicating more molecule detection. The first 4 columns are for the rested condition, experimental condition, and the last 4 are for the activated, going from control to signal muting to the 15-plex and to the enriched T cells. Overall, sensitivity or molecules per cell detected increases when the high expressors were muted or removed. For the enriched, as a note, for the enriched T cell subset, non-T cell markers CD19, CD56, CD11B were not detected, so are in dark blue in the heat map shown. On the right-hand side, we are showing the molecule counts across all the cells for several tertiary markers in active T cells. And we see an increased sensitivity when the high expressors are muted or removed. And this is indicated by a shift in the median molecules detected to the right. When we seek sequencing depth obtained from a given number of reads per cell, and this is shown on the left-hand side, where we have sequencing depth on the y-axis and mean reads per cell on the x, it shows that for the same sequencing depth can be obtained with a 60% reduction in sequencing reads when the high expressors were muted or removed. And this is highlighted by the dotted green line and the green arrow, where we see the same sequencing depth obtained from 10,000 reads per cell for the control experiment in the yellow line versus 4,000 reads per cell for the two experimental conditions, the 15-plex and the signal muting, in the orange and blue line. The sequencing depth shown there in the dotted line is a depth of 1.5. We then compared the 4,000 reads per cell sequencing data from the signal muted sample to the 10,000 reads per cell from the control data in the ability to detect positive cells. And that's what's plotted on the right-hand side. And so as you can see, despite using 60%, despite reducing the reads by 60%, it shows the same performance level to detect positive cells. I do want to say that the 60%, the number of reads required, the 60% reduction, is based on this specific AbSeq panel employed in this experiment and the sequencing depth we wanted to obtain of 1.5. And so then when we further molecule counts across conditions, overall the AbSeq molecules detected from signal muting, which is shown on the left plot, or the 15-plex condition, which is shown on the right plot, we see very high correlation to the control, despite the fact that there is 60% reduction in the sequencing reads used for both the signal muting and 15-plex experiments. TSNE plots derived using both the mRNA and the AbSeq variables. When we do this, we see almost identical performance determined from 60% less reads when the high, and this is for the conditions where the high expressors are either muted or removed. And we see these being consistent between both the resting and the activated conditions. So on the right-hand side, again, the resting condition is in red and the activated is in blue. And we see that there's a difference between those two states, but when you look at the TSNE for the control versus the signal muting condition or the 15-plex condition, you see they are very identical, but are using 60% fewer reads to obtain that data. And importantly, even though the 15-plex, in the 15-plex panel where we removed the high expressors, CD4 was removed. However, CD4 positive T cells were still identified because they were segregated through the WTA data that was also generated in this experiment, suggesting that in order to reduce sequencing costs, some of the high expressors can be removed from your panel design when you're doing this in conjunction with mRNA analysis. For your experiments. Despite less sequencing costs, we believe that we can also provide increased antibody sensitivity for lower expressors, and through that, deeper insights can be derived. So not only can you optimize your panel design to spend less on sequencing, you can actually get increased sensitivity for your lower expressors in your panel. So just to do a deeper dive in this and to look at this, we compared the TS&E plot of the resting and activated PBMCs using both the mRNA and AppSeq variables to generate the TS&Es. The control condition is shown in the top, highlighted in the blue box, and the signal muting condition is shown in the lower TS&E plots, highlighted in the orange box. We then used the different AppSeq markers to visualize different cell lineages and clusters of cells, and that's in the 12 TS&E plots shown. And we looked at CD11B, CD19, CD56, CD25, CD62L, CD197, CD223, CD279, CD357, CD69, CD8, and CD4. When we did that, what we observed was a subset of active T cells identified by a subset of active T cell markers CD279, CD357, CD69, and CD25 only in the signal muted condition in the bottom TS&Es. And this is indicated by a red circle in all of those 12 TS&Es. This small subset highlighted in the red circle in the bottom TS&Es was not segregated in the control condition where the sensitivity for these markers was lower because all of the reads and the molecules were being given to the high expressors. So not only can you save on sequencing costs, you potentially can gain new insights into your biological systems by applying one of these approaches. So in conclusion today are 3 methods to reduce sequencing costs for single-cell multiomics approaches. Muting or removing high-expressing antibodies can redistribute the reads to lower-expressing antibodies, thus increasing the sensitivity to detect low-expressing molecules and improve performance. Enriching your cell type of interest up front can also reduce the cost for markers expressed on other cell types. And also, importantly, through the combination of signal muting for high expressors and also enriching your cell type of interest, you can reduce your sequencing costs and still while obtaining high quality data. And also, importantly, through the combination of signal muting for high expressors and also enriching your cell type of interest, you can reduce your sequencing costs and still while obtaining high quality data. Hi everyone. Before we go to the Q&A, I just want to thank you all for your attendance today at the Ask the Expert webinar. And I also really want to thank the team that contributed to the great work that was presented here by myself, especially Haewon Song, who was the first author on this and did all the experimental design and analysis, Maggie Nakamoto, Punya Narayan, Kathy Lazarick, and Ian Taylor, and also to the members of the Biosciences Immunology Applications team that helped us in the panel design and determining some of the activation conditions used here. I also, before I hand it off for the live Q&A, just want to clarify, there was a small typo on one of the slides. On this slide here, in the signal muting condition, the number of reads mapping to the high expressors is actually not 9%. And if you actually look at that pie chart, you can see it's not 9% of the reads, it's actually 31%. So I just wanted to clarify that. And so now I'll just hand it off to Annalise to kick off the Q&A.
Thanks, Stefanie. So we are now going to start taking questions from the online audience. We had quite a few questions come in, and we'll try to get to as many as possible, but do note that we will respond directly to any unanswered questions by email after the webinar. So for this live Q&A portion of the program, I am pleased to also welcome some additional experts that will be helping to answer your questions. First, as Stefanie mentioned, is Hye-Won Song, who is the first author on the work that was presented. She's a staff scientist with the single-cell multiomics R&D team. And then also joining us are Melissa Perpak, who's a field application scientist for the U.S. region for single-cell multiomics, and also Adam Zubeidi, who is the bioinformatics support engineer, also covering the U.S. region. So welcome to you all. Okay, so I'll get right into the first question, and I'll direct this one to Hye-Won, but other experts, please feel free to jump in when you're ready. So the first question is, what is the difference between signal muting versus using lower amount of antibody, which will save money for the AbSeq reagent as well?
Hi, yes, it's a great question. We believe the signal muting is more controlled way to reduce the molecule numbers detected. If you dilute the antibody, it will show the reduction in quite random way due to the concentration to antibody performance not always linear. So many times antibody activity plateau when you increase the concentration, so that it really makes hard to predict the optimal amount of antibody to see the decrease of molecule. Essentially, we feel it's better to ensure you are always staying in saturation for this region.
And I want to give an opportunity to other experts if they have anything else to add. Okay, I'll move on to the next question, which is, and I'll start by directing this to you also, Hye-Won, but everybody else can also jump in. How do you know the best ratio to mute the signal with purified antibodies?
Yeah, it's another good question. In this experiment, we used the 1-to-2 ratio, so 1 of AbSeq and 2 of the purified antibody. What we actually did was to test a different ratio of muting, so that we did 1-to-1, 1-to-2, 1-to-5, and we measure those by flow proxy to see how much we reduce the molecular detection, and also we don't lose the sensitivity to detect the positive cells. So the flow proxy protocol is also published in our BD website.
Okay, thank you for that. And so somewhat related to that, I'm going to direct this question to Melissa. Does the optimal amount of AbSeq antibody used need to be titrated in advance, or is it already supplied in the appropriate concentration?
It comes supplied in the appropriate concentration. So with an AbSeq vial, you will receive 25 microliters, and you use 2 microliters per sample.
Okay.
So a vial is enough for a few experiments, at least.
Okay. And very interesting question here. I'm just going to throw it out to the panel, and maybe specifically point this one to Stefanie. Just curious how much was saved. My lab has a tight budget.
Yeah, I mean, it's going to depend on your panel and how many high expressors you have in there. But, you know, in this example, we were able to actually use 60% fewer reads. So you're saving, that's directly proportional to your sequencing cost. So, you know, if you had to use, 2 million reads, for example, then you can use 60% fewer, so less than 1 million. So that's directly proportional right there.
Okay, got it. Okay, and another question I'm going to direct to Hye-Won. Are you increasing the molecule count or abundance of low expressors?
Oh, yeah, it's another good question. So we are not really increasing the molecule count or anything. it is actually, we are increasing the chance to capture and sequence the molecule associated with the low expressors. So they are there and bound to the cell is the same amount, but because of the high expressor and the limited number of reads in the experiment, it's lower probability to see and detect those molecules. But now we muted, and then there's a higher probability to actually detect those lower expressors. But the amount of molecule won't change. And also, I think I didn't really answer the question earlier. The detect the best ratio to muting of the signal with the purified antibodies. Actually, I want to add something because I mentioned the flow proxy experiment. It's actually flow cytometry way to quantify the antibody performance of F6. So it's actually related with the oligo probe with a fluorescence probe in it, so that you can use the flow cytometry to measure the antibody performance without actually spend money on the sequencing.
Okay. Thank you. Does anyone else in the panel want to add to those questions before I move on to another one?
I want to add just to clarify that, when you buy the AppSeq reagent, they all come as a conjugated version with the DNA oligo in the right, in that specific test size, and that you do have to purchase the unconjugated antibody and do the titration yourself. We aren't at this time supplying them in that format. So I just wanted to clarify that.
Okay. Good. And then the next question, directing to you, Stefanie, for muting. So I think this person's saying you said use a ratio of 2 to 1, and they're asking, is it 2 tagged versus 1 untagged?
No, it's the ratio that we considered ideal for this experimental system was 2 to 1 unconjugated to conjugated. So you're actually using more of the unconjugated antibody, and Heywan can double check that, but yeah.
Yes, that's correct.
Yeah.
Okay, and then I think this is a related question, but just for clarity, I'm going to ask it. What is the recommended ratio for muted antibodies to non-muted?
Yeah, so what we used here was 2 times more unconjugated versus conjugated. That's what we found was optimal. But as Heywan pointed out, there is a protocol on our website to actually test by flow proxy what would be the ideal ratio where you're muting the signal enough, but not over muting. And so, it might depend on your specific protein panel that you're using. But we can, a good guideline is to probably test 1 to 1, 2 to 1, and then maybe 3 or 4 to 1, something in that ballpark.
Okay. All right, and the next question, and I'm going to start by directing this to Heywan. What is the dynamic range of protein versus mRNA and recommended sequencing depth for antibody compared to mRNA?
Okay, so the mRNA copy number is actually quite, the dynamic range is much lower than protein. So protein abundance stretches over at least 7 orders of magnitude, from 1 copy per cell to 10 million copies per cell, while the mRNA dynamic range covers only 3 or 4 orders of magnitude, so that the protein has a higher range. That is why we actually come up with this method to help the customers. And so we recommend for the mRNA on the WTA type of experiment, we typically recommend 5 to 10,000 reads per cell. It depending on the cell type of interest as well. So some cells doesn't have high abundance of RNA. Some cell has really abundant mRNA. So that you have to read more deep to get the full information. And for AppSeq, we normally recommend 500 to 1,000 reads per antibody per cell. So, example, if you have a 20 different antibodies, then you need a 20,000 reads per cell without signal muting. And with the signal muting in this experiment, actually we could actually go down to 200 reads per AppSeq per cell, and then we could detect almost same results as a 500 reads per AppSeq per cell. But it really depending on your panel design, your experiment design, and also your sample of interest.
Okay. Does anyone else on the panel, any of our other experts, want to add to that? Okay. So I'll move on to another question. This is, I'll start by pointing to Stefanie, and then if anyone else can jump in as well. But the question is, can't you just remove the high expressors in your panel rather than muting?
So yes, absolutely. And that was the condition in this presentation where we just labeled it 15-plex. So the total panel was 20. 5 of those were the high expressors. And so the 15-plex is just taking those out. And so absolutely you can do that. And the nice thing is that, you know, as we mentioned, you can still detect, if you have the mRNA in there, you can still detect those cell populations, even if you, like we showed with the CD4 positive T cells, if you have the RNA to help with the segregation.
Okay. Right, but certain cases, actually, the high expressors are absolutely required. In that case, actually, you want to mute. So certain cases, you have a big dynamic range of those high expressor, and the expression level differences really matter for your experiment, so that you really want to have that expressor. Then you actually come up with this muting approaches.
Okay. And going to get to a second question here and point this one to Melissa. Is the recommended peak depth still the same after signal muting?
I believe that this. Stefanie?
Yep. You want me to take that one? Yeah.
Please.
Okay. So yeah, absolutely. So the sequencing depth is how many reads you're getting per molecule. And so, you know, if someone might want a sequencing depth of 1.5, someone might want to sequence really deeply, get a sequencing depth of 3. So yes, absolutely, that target stays the same. What we're showing here is a way to get that by using fewer reads.
Okay. All right. And then also another question, is the only gain a cost reduction, or does it improve the data or interpretation? I think maybe Stefanie, do you want to start, and then anyone else can respond?
Sure. Let me start, and then we can see if anyone wants to follow up. So the primary goal of this was to just reduce the cost. We didn't want people unnecessarily sequencing deeper than they need to get the data on all of the specificities in their panel. So that was the first intent. I think you can, in theory, use this method to actually get more uniform and slightly deeper sequencing on your system to potentially, as I alluded to at the end there, get more data or deeper data on your experimental system. But that wasn't the primary goal. The primary goal was to actually say, hey, you don't have to sequence over and over those high expressors, right? You just need to sequence those deep enough, and it's more important for most people to actually get the reads attributed to those lower expressors that may be giving them more new information or more insights.
Okay. Anybody else want to add to that? No? Okay. So this question, I'm going to start with you, Melissa, and then again, anyone else can jump in. Do you know the sequence of the RNA after sequencing? Do you use that information to analyze RNA?
Sorry, I'm having audio problems.
Sure, I'll say the question again.
Or maybe Adam can.
Sure.
Yeah,
Adam,
I think I can address this one.
Okay.
Yeah. So again, I'll repeat the question just in case anyone missed it. It was, do you know the sequence of the mRNA after sequencing? And can you use that information to analyze RNA variation? Or can you only know which proteins are related? So we do know the sequence of the mRNA after sequencing with our single cell experiments on the Rhapsody. We always include a targeted or a WTA experiment alongside our AbSeq experiments. So we are able to get mRNA information that you can then correlate with the protein information. Now, in regards to whether you can detect the variations of mRNA, depending on how close your mRNA is or your particular gene is to the 3' end of your transcript, that is a possibility. We do have options, again, depending on the location of that gene, to try and create an experiment to detect that. And if that was something you're interested in, we definitely encourage you to reach out to us and we have a chat with you about that.
Okay. Thank you for that. Okay, so another question here is, what is the what is the detection sensitivity difference between oligo-Ab in your workflow and fluorophore-Ab in traditional flow cytometry? Maybe I'll start with Heywan, and anyone else can jump in on that. Let me know if I should repeat the question.
Okay. Yeah, great question. Fluorophore antibody is actually amplifying the signal due to the fluorophore. So depending on the dye, actually the sensitivity can vary a lot. Oligoantibody in the other way, we actually don't amplify. It's just giving us the true molecule number. Depending on which kind of dye your choice, and in my personal experience, I think the detection sensitivity itself is not really not much different. The big merit of FACSMatters is that we don't have much of the background. All of the fluorescent antibody has a background, so we'll see that much of background ticks, and it's sometimes hard to distinguish real signal versus the background. But FACSMatters is almost like zero molecule background, so that we will see the detection of as low as possible. You can actually see the data and can take as a molecule number. Hope it's answered the question.
Yeah, and if I could add on to that, in addition to Heywan's answer, all of our AbSeq antibodies go through a rigorous QA process to ensure that they're all concordant with their flow counterparts. So all the AbSeq that are available in our catalog today have concordance data that we would be happy to talk about with you if that was something that you were concerned about. But if they are not concordant, if they're not concordant enough with our flow antibodies, then we simply don't release them. So you can be assured that every one of these AbSeq oligos will have similar performance to the antibodies that you're used to using in a flow setting.
Okay. All right, thank you. And so another question, I'm going to start with you again, Heywan, here. Do you see better results if you dampen signal by using unconjugated AbSeq antibodies versus if you simply lower the concentration of those same AbSeq antibodies in your stain mix?
also I think it's the same, almost like similar question as the first one. So, signal muting versus the titrate down the amount of antibody, right? Yes. So we believe we actually titrated. Actually, when we released the antibody, we have all the data in different titration of antibody concentration. So we titrated and we did experiment, and then we realized what is the optimal concentration to the saturation of the staining, and then we released that optimal concentration for the individual F-stick reagent. It's all optimized in that way. So we know the titration curve, how it looks like. And normally it doesn't really show us the linear range of the titration. So it will be really hard to come up with the concentration will work as we desired for the signal dampening. And if we choose to titrate down the antibody concentration. So rather than that, this combination of the unconjugated antibody mixed with a conjugated antibody actually showing that very controlled way to reduce the molecular number, and it's all very well correlated and predicted. So we believe it's a much better way. And also, when you titrate down antibody, it's not near the saturation. So when you want to do the antibody staining, you want to have a saturated antibody in your staining. Otherwise, you will get some uneven staining and also can have some bias detection. So we believe the signal muting is a better way than the titrate.
Okay, thank you. And so we're gonna wrap up our Q&A now, but this is the final question, which I'm gonna direct to Stefanie, and it's really simple. Someone wants to know what instrument was used for sequencing.
Yeah, our right now all our sequencing libraries can only be sequenced on Illumina sequencers. So I believe we probably used a NextSeq 500 or 550 for this one.
Okay, perfect. Well, that is the last question for the webinar. Thank you all so much for joining. Note that CE credits will be given to attendees, so look out for an email with more details. And also a recording of the presentation will be available on the webinars playlist of the BD Biosciences YouTube channel, as well as on the webinars page at bdbiosciences.com. And with that, thank you for joining. We hope you found this presentation useful, and have a great day.